Hey everybody, and welcome back to Microbiology Lecture. We are into Chapter 8, Bacterial Genetics. I'm your host, Professor Mary Annarella, and we're going to get into some cool stuff here. And bacterial genetics meaning inheritance of traits from one bacterial cell to another. So just to give you a little bit of background, Staph aureus, or S. aureus. It's a bacterium that's commonly found on skin, and it's often the cause of wound infections. With the advent of antibiotics have been a game changer with public and personal health. Prior to the advent of antibiotics, and I want to say it was like the late 40s, around the late 40s when the use of penicillin came into use, people used to die of infections like, from strep throat even. Helen Keller, for example, was blinded and deafened as an infant because she had scarlet fever. That's caused by a streptococci bacterium. That wouldn't happen today because a child with that sort of infection would be treated with antibiotics. But it was around 1954. I want to say it was around 1954 when we began seeing resistance from Staph aureus to penicillin, for example. So antibiotic resistance. And several Staph aureus strains began developing the ability. They had a protein to basically break apart the beta-lactam ring in penicillin and rendering it resistant to penicillin. So we couldn't use penicillin to treat Staph aureus infections anymore. Since then, we're constantly developing new antibiotics because bacteria keep developing resistance to it. And it's a huge problem, like MRSA, methicillin-resistant Staph aureus is a huge problem in the medical community, in hospitals. Methicillin is a form of penicillin that's only used in the hospital, and it's like a last-ditch effort. So antibiotic resistance is a huge problem. How do bacterial communities develop antibiotic resistance? In two ways. Natural selection. If there is one resistant microbe around in an infection when you're treating it with a particular antibiotic, that one's not going to be killed by the antibiotic, and it's going to divide and pass on more of its resistance genes onto daughter cells. So natural resistance and basically what we call vertical inheritance. Vertical inheritance is passing your genes on to daughter cells. Passing genes on to daughter cells. So, in eukaryotes, that would be by mitosis, and in prokaryotes, that would be by binary fission. Or you could have a mutation. Mutations to genes, I want to say, are like one of the major, I want to say, like, I read somewhere about 50% of antibiotic resistance are caused by mutations to genes. And mutations can happen spontaneously, or they can be induced. So we're going to talk about a few of those things. So first, to talk about mutation. When I'm talking about mutation, let's say we have a gene here. And it's, I don't know, blah, 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 GATCATG, CAC GTTACC, blah, blah, blah. This strand is going to be transcribed into messenger RNA. So the messenger RNA is going to have, it's going to be analogous to this. So GAUCA, oops, AUG, CAC, GUU, ACC, blah, blah, blah. Don't worry about wrapping your head around, well, wait, isn't it supposed to be complementary to this? I didn't draw the other strand that this transcript was transcribed from. So let's just look at this. Okay, so here's the first AUG, and you know that that's going to lead to methionine, right? So this ATG is actually analogous to the start codon AUG. Okay, but let's say when this piece of DNA was copied back here, let's say that a copy was made, but DNA polymerase made a mistake and it got a little confused and it saw, it was making a copy and it made ATG, CAC, GTT, and then it sees an A and it accidentally added one extra T. Can you see that the messenger RNA then is going to have a U in it? That's going to be a U. So when the ribosome is reading this right here, you're going to have AUG, which is going to be methionine, CAC, I forget what that is. Let me look that up. Oh, you get to see all my bread pictures. Great. Here we go. CAC is going to be, CAC will be histidine. Histidine, here we go. GUU, what is that? GUU, GUU is going to be valine. I'll just put val for valine and that little dehydration reaction will occur. Okay, now the next codon was supposed to be ACC, but now it's UCC, and that's going to give us two different amino acids, ACC and UCC. ACC is threonine and UCC is serine. All right, that's a mistake, and you're going to get, instead of threonine, we're going to get a serine. So this was going to be threonine, but that mistake makes it a serine. That may or may not make a difference in the protein, or it might change everything. So for example, in sickle cell anemia, there is one base change that changes the amino acid that causes the protein to be totally different and ends up causing sickling of red blood cells. And the same sort of stuff can happen, for any gene. So let's take a look. Here we go. I want to look at, okay, what happens when a mutation is introduced to the DNA? Maybe nothing. Because the code is redundant, maybe you won't get a different protein. Or maybe you will get a different amino acid and maybe it'll mess up the protein. Maybe it'll create a protein that gives the microbe superpowers like antibiotic resistance. That's the stuff that we're interested in there. So let's see, I already talked about antibiotic resistance in Staph aureus. It's a huge problem across the board with all microbes. All right, we get it through natural selection, previously resistant, or mutation. So let's talk about the types of mutations. There are. So types, we definitely need a different color here. Types of mutations. Because DNA polymerase sometimes makes mistakes. Mistakes get through. When you're copying something word for word, people still make mistakes. So it's the same thing because DNA polymerase sometimes makes mistakes. Haha. S-I-C. S-I-C means I deliberately misspelled that. I did that on purpose. All right. So you can get different types of spontaneous mutations. One is called, that I already showed you, is called a base substitution. A base substitution is like right here. DNA polymerase was supposed to put an A there, and instead of put a T, it got a little confused. So the wrong base nucleotide gets added. If it's only called a base substitution, this is also known, rather I should say, if it's just one base substitution, it's called a point mutation. So a base substitution is also known as a point mutation. If that point mutation causes a change in the amino acid, it's called a missense mutation. You get a different amino acid. If that point mutation results in a stop codon, this is called a nonsense mutation. A nonsense mutation results in the creation of a stop codon. I think it's UAA or something is one of the stop codons. Yeah. But wait, that's not all. There are different types of mutations other than a base substitution or point mutation. Let's take a look at what that would be. So let's come back up here. Okay, so let's say DNA polymerase is going along its merry way. It adds an ATG, CAC, GTT, oh, T, ACC. Let's say it put in an extra T right there. So when the messenger RNA is created, it's going to have an AUG, CAC, GUU, UACC. There's going to be a U in there, right? So when the ribosome reads the codons, it will first read AUG, CAC, which is histidine, GUU, which is valine, but the next codon isn't going to be ACC. It's going to be UAC. Guaranteed that's not serine or threonine. It was supposed to be threonine. But let's look up what UAC is. UAC is tyrosine. Tyrosine. But more than that, can you see, let's say I'm going to go on and put an ACCGTT in there, and this will be ACCGUU. Can you see that everything is going to be off now? Like these were supposed to be the codons here, ACC, ACCGTT, and instead it's UAC, CAC, CGU. Can you see we're totally off there? Everything is going to be messed up from that point on. Okay, this is called a frame shift mutation, and it's when either an extra nucleotide gets added or one less. Like what if it skipped over that T? That would do the same thing. So let's call this add or remove a base or nucleotide. This shifts the reading frame. It changes the reading frame. So it's called a frame shift mutation. And basically, every point after the added or deleted base, the extra base or the one less base, it's going to screw up the codons totally, which means it's going to screw up the protein from that point afterwards. And just to point out one more little thing about a reading frame, let's say we have a piece of DNA here. Can you see if we have A, T, C, A, T, G, A, G, U, A, C, A. Nope, I shouldn't put a U in there. What am I doing? I should have gone with T. T, 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 A, C, A, G, blah, blah, blah. Can you see that? I'm going to add one more over here. There we go. Can you see that we could group the codons like this? Or we could read them in groups of 3 like this. Or we could read them in groups like this. TCA, GTT, da da da. These are called reading frames, and there are 3 different reading frames. Now, what determines the reading frame of a particular gene is that first ATG, because that's going to mean AUG in the messenger RNA, and that's the start codon, right? So the ATG in the DNA, which is the AUG in RNA, determines the reading frame. It determines the reading frame. And if you pop in an extra base here, or if we get rid of that G, that's going to change the reading frame from this point on. From that point on. I think my stylus is running out of juice here. And I think my iPad is as well. So you get a reading frame, you get a frame shift, which definitely changes things. There is a third way that mutations can be introduced into an organism, like on its own, or we could do that in the laboratory too. What are we up to here? We got 2. I'm going to go back to yellow. And that is through the use of what we call transposable elements. I'm not sure where my pen isn't working so well right now. Transposable elements, also known as transposons. Transposons were actually discovered and studied by a woman named Barbara McClintock. I'm not sure if I'm spelling her name correctly, but she won the Nobel in 1983. I remember that. I remember hearing that she won. She worked at a laboratory in Cold Spring Harbor, and she studied transposable elements in corn. So she first noticed these in corn, and what transposable elements, or also known as transposons, also known as, one more AKA, jumping genes. Transposable elements are DNA sequences that can cut themselves out of a chromosome all by themselves and reinsert themselves someplace else. It's really wild. When she was studying corn, I forget how many chromosomes are in the corn genome. I don't know. But she noticed that if this is a corn chromosome, I'm just kind of drawing it like a wavy line here, and there was a gene that she was looking at, she was able to detect it to a particular location on, I don't know, let's say corn chromosome 9. She would breed the corn, and then look at basically the baby corn and find that in the next generation, here's chromosome 9, she found that it moved to chromosome, I don't know, I'm making up this number, I don't know this number for sure, but let's just say moved to a different location on chromosome 6, and that it wasn't there anymore on chromosome 9. It had moved, it had jumped. Nobody understood her work when she was doing this, like back in the, I want to say it was the late 70s. No, it would have been early 70s. No idea. Whenever she was doing her work, people just really didn't understand it. So, they didn't think she was crazy. They just didn't understand the work because, whoa, this was so far ahead of our understanding in how inheritance happens and the expression of genes. We thought the location of genes were permanent on chromosomes, but they can actually move. And we can actually use these transposable elements now to do this on purpose. So for example, we can do something called knockout mutations. We can create something called a knockout mutation by using the sequences of transposable elements and trying to get them to insert into a particular chromosome of, let's say, a bacterium in culture, for example. And can you see that if we have these transposable elements here, let's say we're studying something in mice, and this is, part of the mouse DNA, and there is a gene here. Let's say we take a transposable element, transposon, right here, and we get it to insert itself somewhere in the mouse genome, and this thing ends up looking like this. Oops. We popped it right in the middle of this gene here. Can you see that this gene, I don't know, gene X, is now going to be inactive? We just knocked it out. It is a knockout mutation. This can be a very helpful research tool. So if we don't know what the function of gene X is. So for example, a study was done, 30 years ago in mice, where they were looking at gene X was the gene for P53 in mice, which you may recall is actually involved in taking cells out of the cell cycle when they are when they're too damaged, when they've damaged DNA. And here's what we found. Here's what they found. Where's my mouse? No, that is not it. Here we go. So I'm jumping ahead several slides here because I think this is pertinent. All right, so we didn't know what the function of P53 was until we created some mice that where we had knocked out the gene. So I'm going to have to take a picture of this because I got to draw on it for you. Come on. All right. What this plus sign here means, this plus, Plus means a normal copy of P53 gene. So this mouse here has 2 normal copies of the P53 gene. This mouse here, rather I should say these mice have 1 good copy, but 1 knocked out copy that was knocked out with transposons. This mouse down here has 2 knocked out copies. So these are P53 knockout mice right here. Okay, the mice up here with 2 good copies of P53 developed no tumors. This is actually breast tumors that they were looking at. Only 1% of these mice who have, with 2 normal copies of their P53 gene, only 1% of them developed breast tumors at 18 months. In the heterozygous ones here, only 2% at 9 months. The P53 double knockout mice, 75% of them develop breast tumors by 6 months. So this informed us that, oh, if a mouse lacks 2 copies of their P53 gene, they're going to get tumors. So therefore, P53 is a tumor suppressor gene. They used knockouts and transposable elements to help determine that. So it was a helpful thing to know, and now we know what P53 does, and we actually have learned a lot more about it now. So there we go. Okay, so let's come back here. Okay. Types of mutations. We got base substitutions, also known as point mutations, missense, and nonsense mutations. Frame shift mutations will result if you add or remove a base, and transposons can essentially be a knockout mutation because they can knock out a gene. We do have ways, however, of inducing mutations. Like, okay, if you're, well, actually, we did use transposons to induce a mutation, but it takes forever. If you're waiting around for a base substitution to happen, in a microbe for your research, you're probably going to wait a long time. If you're studying plants, for example, you're just going to wait a really long time for a particular mutation to occur. We have known for a long time that chemicals can cause cancer. A lot of chemicals cause cancer by altering DNA. And so that is why certain chemicals are labeled a mutagen or a carcinogen, because they cause mutations that can lead to cancer. So there are 3 different ways to induce mutations. This first one, we'll talk about chemicals. Transposition is using transposons. Already talked about that. And then third, irradiation. So let's talk about chemicals next. Okay, so let's see, inducing mutations. One way is through using chemicals, and a chemical mutagen. Oh, my Apple Pencil battery is low. Oh no. I'll plug it in for just a second here. But chemicals that induce mutations, we call them a mutagen. If it's likely that those mutations lead to cancer, and generally they do, most mutagens are going to be what we call carcinogens. Right. So there are different ways. Come on. I'm plugging this in. All right, we're going to have to take a break here, folks, because I am noticing that not only is my Apple Pencil battery low, but my iPad needs to be plugged in, so I'm going to go take a minute, and there's no reason for you to have to sit through that. Okay, we're back. I've charged my Apple Pencil a little bit, and the iPad is all plugged in, so let's get back to talking about chemicals. Woohoo! All right, there are different types of chemicals that are mutagens. Different classes of them. One class will actually mess with the DNA that is already there. It will chemically change the base portion of a nucleotide. So let's say chemicals that alter DNA bases that are part of existing DNA. It'll chemically change a G, A, T, or C, and it will alter its hydrogen bonding pattern. And what you get from these are base substitutions. One type of these are alkylating agents. They add alkyl groups to DNA. And smoking, smoke apparently has a lot of alkylating agents, and so one of the common people who study lung cancer, for example, sequence the DNA from lung cells from patients compared to healthy lungs, smokers and non-smokers. And there's some interesting data that I thought I'd show you because you do get some base substitutions from this. And here we go. Where are we? It was right after the mouse. There we go. Okay, so, wait a minute. Even though this is actually in PowerPoint, you're not getting the little animation thing that I have here for you. Let's see. Easier if I take a screenshot. So this is looking at specific mutation pattern in lung cancer from smokers, particularly looking at things that were supposed to be a G but have been base substituted into a T. You have 30% of the tumors from lung cancer patients who are smokers have that G to T base substitution pattern compared to that of non-smokers and non-tobacco related tumors. So that's a pretty big difference. At a greater proportion, smokers have a greater proportion of G to T base substitutions because smoke does contain carcinogens, and apparently a lot of them are types of alkyl agents that alter the DNA in your lung cells and result in base substitutions. There we go. Another type is what's called base analogs. Base analogs. An analog is something that looks like but isn't quite similar to the original thing. So over here is the normal nitrogenous base. Over here is the analog. Here's a thymine. This is what thymine looks like. 5-bromouracil replaces that methyl group right there and has a bromine on it. This one here, a regular adenine. The ammonium group gets moved from here down to here. Here we go. We have, oops, come on. There we go. We have thymine over here. Put a T on the thymine because it's the sugar part of the thymine that gets messed up here. This is the sugar part down here. The thymine would be up here. There's no change between the two. AZT changes it to 3 nitrogens from the OH group, and that's obviously going to interfere with forming with the next nucleotide adding because the whole nucleotide adds on here where you have the sugar phosphate backbone thing going on. Not going to happen here. AZT actually was the first somewhat effective AIDS drug. It's not used anymore because protease inhibitors work so much better, but an early AIDS drug was AZT, and what it did was it caused the virus to incorporate this base analog into it, the HIV, into it, and slow the growth of the virus itself. Unfortunately, HIV mutates very rapidly, and this drug became useless against it. Later in the course, we'll actually talk more about what's used to treat an HIV infection. Anywho, so we got to get back to this part here. Chemicals. Chemicals that alter existing DNA. Another type of what we just talked about are base analogs. They're something that look like a base and get incorporated into the DNA and mess up its hydrogen bonding properties. So they are nucleotide lookalikes. They think of them that way. Nucleotide lookalikes that get incorporated into DNA. So this is for actively growing cells. These chemicals up here, these mess up DNA that's already in existence. Base analogs will mess up DNA that's being copied, and they get incorporated into actively copied DNA. Actively replicating DNA, that's the term I want to use. Right there. A third type is called an intercalating agent. Some chemicals are what we call intercalating agents. I've heard some people call them intercalators. I think intercalating is the way it's actually supposed to be pronounced, but I don't really care. But an intercalating agent works differently. It doesn't get incorporated into DNA. It doesn't mess up a base that's already in there. But if you have, like, let's say here's a piece of DNA, GATTCAGCATGCCACCC, an intercalating agent is like a physical roadblock. I'll use this. And it will insert itself and attach to the DNA. And it'll just sit there at certain spots, so that when DNA polymerase comes through to make a copy of it, and it's like, oh, I'm just zipping along, adding the new 5' end first, and I'm going to put, let's see, a C, T, A, A, G, T, C, G. It'll see this roadblock and go, I don't know what that base is, but I don't know, maybe I'll put another G, something like that. T, A, C, G, G. I don't know what this base is, but maybe I'll just add another G. Intercalating agents cause frame shifts. So you get frame shift mutations. An example of this is a substance called ethidium bromide. Ethidium bromide will intercalate itself amongst the DNA, and actually when you shine UV light on it, it makes the DNA glow. It glows so you can see where the DNA is. We actually use this in the laboratory to be able to see DNA on a gel electrophoresis. We don't use it at HCC because it's a carcinogen. It will totally screw up your DNA if you get it onto your fingers. It'll be absorbed into your skin. You don't want to use that. Ethidium bromide, it is a carcinogen. It's a carcinogen and it's an intercalating agent. It'll mess up DNA. Okay, so let's see. You can induce mutations, I guess let's put an A there, through chemicals. You can do them through transposons, which we already talked about, doing a knockout mutation. Or you can mess up you can induce mutations a third way, and that is with irradiation. We talked a teeny little bit about this when we talked about this in Chapter 5, the control of microbial growth. And we often, for example, irradiate food to sterilize it because the irradiation kills the bacteria by mutating the bacteria's DNA. Okay, now this only works for actively growing bacteria, and endospores are resistant, but X-rays, for example, X-rays and actually gamma rays, these will introduce double-stranded and single-stranded cuts into the DNA, and it'll result in a deletion of bases, so you get a frame shift. So introduces, is this? I did not, apparently, And my pencil's going bad again. All right, so I'll talk at you for a second. It just takes a couple seconds to charge up the pencil, enough for me to write another paragraph. But X-rays and gamma rays introduce double-stranded and single-stranded breaks, cuts into the DNA. UV irradiation works a little differently. So make sure you have it in your notes that X-rays and gamma rays introduce double-stranded. This is not going to work. I'm going to have to come back in a few more minutes. All right, we'll see if 10 minutes was enough to charge my Apple Pencil. I don't know, there might be something going on with it. Anywho, X-rays and gamma rays. Yep. There we go. Introduce double-stranded or single-stranded cuts into the DNA. Whereas UV light does something different. UV light causes what we call thymine dimers. Not dimmers, but thymine dimers. Thymine dimers occur in the presence of UV light on a piece of DNA, like let's say we have GATC, ATT, CACC. This, okay, something's up with my pencil. ACC, blah, blah, blah. And what thymine dimers do is cause, I'm sorry, I'm just really distracted by this pencil not working. Why is it not working? CAC, do something like that. What it does is it causes a covalent bond to form between two adjacent thymines. So you know that these bases are hanging off of the sugar phosphate backbone. And they're only attached to the sugar phosphate backbone. They're not attached to each other like here. Not at all. A thymine dimer will occur in the presence of UV light. UV light causes two adjacent thymines to basically covalently bond to each other. So it's a covalent bond between adjacent, that means right next to each other, thymines. And it's like a kink in your zipper. You know, did you ever have your zipper, like imagine if two teeth of your zipper got glued to each other. That zipper's not going to zip past that. Yeah, so what you do is you get base substitutions. And you can even get a frame shift if it just adds one base instead of two there. So UV light works a little differently from X-rays and gamma rays. What's going on here? Thymine dimers cause a covalent bond to form between adjacent T's. It does not form covalent bonds between non-adjacent T's. Like you're not going to get one from this one to this one over here. That's not going to happen. Nope. So they have to be adjacent. They have to be right next to each other. So that's the scoop right there. And I'm going to see if I can just, oh, all right. Maybe this thing has healed itself, but I'll keep going and get into, okay, I don't want to freak anybody out here with all these mutations that can happen. Oh my God, all the time. Mutations do happen a lot of the time, but there are processes in place that repair them. So repairing mistakes. So first up, your average garden variety mismatches will be repaired easily by DNA polymerase as it is replicating the DNA. We actually say that DNA polymerase has proofreading ability. Okay, so far so good with the pencil. DNA polymerase has a nice proofreading ability in that if it sees that it's going along and it sees a G and it adds a C, okay, it's happening again. It's happening again. It's happening because I didn't have this on here. Okay, let's see. There we go. Me and my neuroses. All right, it sees a G, it adds a C. It sees an A, it adds a T. It sees a T, it adds an A. Blah, 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 blah. And let's say it does that and then does this and goes, whoa, whoa, whoops, whoops. I've noticed that this is a mismatch. It can actually back up and fix it due to its proofreading ability and get it right. It has proofreading ability. Now here's the deal. You know that DNA is double-stranded, and let's say, okay, DNA replication is going along, 3' and 5' ends here, and the new end, I'll put newly synthesize this leading strand right here. Here's the new strand. Let's say DNA polymerase notices that there is some sort of mistake somewhere in here. How does it know if this strand is correct or if that strand is correct? It needs to know. And here is how it knows. It knows through the process of methylation. Did I spell methyl correctly? Yes, methylation. A methyl group is just a little CH3 group like that. And the longer DNA is hanging out in the cell, it's going to be methylated. Like this. So if this red strand is the original strand, it's going to have methyl groups on it. And this yellow strand here has to be around in the cell a while in order for it to be methylated. So DNA polymerase assumes that the correct base in here, that the correct base is on the methylated strand because it knows that the methylated strand is the oldest one. So methylation determines which strand the DNA polymerase is going to decide must be correct because the more heavily methylated strand is the original strand. It's older. It's going to be a while before this leading strand, this new stuff, gets any methyl groups on it. Has to be around in the cell. And incidentally, methylation has something to do with gene regulation in eukaryotes as well. So what's interesting is that the DNA proofreading process is related to the gene regulation process. Isn't that interesting? All right, but let's say DNA polymerase, okay, it proofread as well as it could. You know, if you've ever had anybody edit a paper for you, they still might accidentally miss a mistake. The cell has a series of enzymes that can do what's called mismatch repair. Mismatch repair. So this is where, if there is a mistake, like let's put in GATTCA, let's make this easy, CCCGGGACA on one strand, and the other strand's got CTAAGGGCCCTGT, blah, blah, blah. We've got a mismatch right here. A does not pair with A, right? An enzyme called an endonuclease will come out and cut the mistake out. So an endonuclease, it's an enzyme, an endonuclease cuts out one strand. I want to erase that here. The one strand with the mistake. Now, Is this strand the mistake or is that strand the mistake? Again, it looks to see which one is more highly methylated. Right, and if all those methyl groups are over here, there are more methyl groups on that strand, the endonuclease will cut out just a little bit of one strand. Like that. So it has cut this piece out right there. One strand, not both strands. And it doesn't cut out the whole strand of the chromosome, just, 5 or 6 bases worth of mistake. So an endonuclease cuts out the one strand with cuts out a piece of the one strand with the mistake. Then DNA polymerase can come back in. It's got 2 strands to bind to, and it can come back and give it another go. Something like that. So DNA polymerase resynthesizes, but you're going to have a little gap here, right? So you need DNA ligase to join the two together. So then DNA ligase seals the gaps. It minds the gaps. And this is just the general way that the cell deals with mutated DNA. It figures out which strand, it sees a mismatch. It figures out which strand is the newer one because it will have fewer methyl groups on there, cuts out a little piece of that new strand. DNA polymerase resynthesizes, fills that in, and DNA ligase seals in those gaps. Now, repairing thymine dimers is a little different. So let's get a whole different color here. Need a different color palette. Let's go with, I don't know. No, we haven't done blue for a little while. Let's do blue. Repairing thymine dimers. Anybody hear my dog snore there? Poor little thing. It's a rainy day here. So she's just sleeping all afternoon. She's an older dog. That's what they do. All right. Repairing thymine dimers. Remember, this is a covalent bond between 2 adjacent thymines. The simplest way to deal with this is that E. coli has what's called light repair or photoreactivation. Okay. That's when, in the presence of light, a special enzyme will come along and just break that covalent bond. So in the presence of light, that's why it's called photoreactivation. An enzyme, a special enzyme, it doesn't have a specific name, will break the covalent bond. Sometimes it's called light repair, photoreactivation. There is what's called, in the absence of light, excision repair will occur, which is the same as mismatch repair. It's just a different endonuclease. So mismatch repair up here, this process where an endonuclease cuts out the strand, only this time it cuts out the strand with the thymine dimer. And then DNA polymerase will fill it in, DNA ligase joins everything together. So same as mismatch repair, but with a different endonuclease. And then finally, an excision repair if no light is around. If there's no light, it's got to do excision repair. And then finally, if there is so much damage, if there is so much UV damage, because UV irradiation is what causes those thymine dimers, if there is so much damage, that's too much for photoreactivation for one little enzyme there, or even excision repair to handle. If there is a ton of damage, what happens is that a whole new operon of genes is activated, and that's called the SOS repair system. It's a whole new operon of genes that includes a new endonuclease and a different DNA polymerase. But the deal with this different DNA polymerase is that this different particular DNA polymerase has a really crappy proofreading ability. So it's a crappy proofreader. I personally am a crappy proofreader. I can't proofread my own stuff. I don't catch all the mistakes. And likewise, this DNA polymerase here, it's a crappy proofreader. It will take all kinds of mismatches. It'll take all kinds of misspellings, run-on sentences. It'll take all kinds of mistakes. But it's a last-ditch effort for E. coli to try and overcome a ton of thymine dimer damage from UV irradiation. And now that my Apple Pencil seems to have totally run out of juice, and I sure hope I don't have to get a new one. I'm going to sign off, and we'll see you next time. We'll see you in class. Take it easy and have a great day.